Scientists uncovered mercury in mongoose brains, indicating a broader environmental threat and raising concern about human exposure to mercury. Exposure to mercury is extremely neurotoxic in most chemical forms. Even scientists who study mercury compounds are at risk due to potential exposure to Hg. Renowned physicist Michael Faraday suffered from Hg poisoning due to prolonged exposure to Hg vapors, leading him to halt his research at the age of 49 due to deteriorating health. Another example is lab chemist Karan Waterhan, who was killed by dimethyl mercury poisoning after a few drops escaped from a pipette and landed on of her latex gloves hand. Numerous studies have focused on the exposure and effects of Hg, that means mercury, particularly in marine and sea creatures. It is well known that people should limit the consumption of certain fish like tuna due to mercury presence. However, the question arises, can mercury ions reach the brains of terrestrial animals? Dr. Yulia Puskar, a professor of physics and astronomy in Purdue University College of Science, was initially skeptical. She has maintained a brain imaging program since 2008 at Purdue University. Her group, with expertise in sample preparation, measurements and data analysis, is sought after by researchers in the US and worldwide, including this from Japan and more recently Australia. Puskar's research group was tasked with checking for Hg in the brains of mongooses collected on Okinawa Island. Surprisingly, brain scans revealed mercury in these invasive animals. The research group refined the scans, achieving a resolution of a few tens of nanometers to observe the affected brain cells. Their collaborative findings were recently published in the journal Environmental Chemistry Letters. The mystery of how mercury enters the mongoose brain remains unresolved. Possible sources include they drink water from their entry, birds' eggs they consume, mineral exposure, or even the air they breathe. One thing is very clear, this is a very bad sign. Hg, that means mercury, is very toxic at low concentration, as Hg can bind and affect the function of essential biomolecules, explains Puskar. Efficiency of detoxification will depend on uptake and binding constant inside detected accumulations and potential leakage from this if brain cells die. As of now, there is no known way to safely dissolve these aggregates from tissue, and there are no reports of reversing Hg poisoning of neural system. The main approach we should all take is to avoid any exposures, especially chronic ones like in Faraday's case. Researchers told, I was skeptical whether any Hg could be detected. Usually neurotoxic elements, even if they get into brains, are present in ultra low concentration, explained Puskar. We took these specimens to the advanced photon source at Argonne National Laboratory, where brains were exposed to intense X-rays, defying my skepticism. The Hg signal was present. Bien, the report.